Good morning, everyone. Welcome to our talk series on the cerebellum. Today we have as our guest Mark Wagner. Mark was an undergrad in bioengineering with Marie Smith at Harvard, where he studied reaching movements in humans and introduced a number of concepts that he continued when he went to Stanford to do his PhD in neuroscience with Mark Schnitzer. He developed manipulandum for mice and began recording from granule cells, discovering that they responded to both omission and anticipation of reward signals. Then, as a postdoc with Li Kuan Lu at Stanford, he discovered that climbing fibers change their way that they respond to movements as the animal learned the task, becoming more synchronized. And then he received his independent lab. He was started at NIH, where he's been for about 2 years. Mark, is that correct? So it's wonderful to have you, Mark. Thank you so much for your time, and look forward to your presentation.
Thanks a lot, Reza. It's always great to be able to speak to this audience, and thank you for keeping this seminar series going. So I'm going to tell you about this story that I've titled "Linking Temporally Distant Events in the Cerebellum," which will finally, after many attempts, be coming out over the next couple weeks. And to kind of dive in, the general motivation is, we at least, I think, like a lot of people, have been animated by these findings from the human literature, right? That there's this very large and I would say growing list of behaviors that seem to have cerebellar contributions, not only classical sensory motor, but all the way up to more abstract tasks like, maintaining things in memory and verbal fluency and these kinds of things. As interesting as this list of behaviors is, I think what we know about cerebellar microcircuit computation still comes from a much more restricted class of behaviors that I would generally call sort of short latency associative learning, right? That there's some predictor that you want to use to synchronize some action to some event. Say, use a light to predictively close your eye before you get hit in the face with some air. And we know a lot about the cellular underpinnings of this type of learning, right? That predictive information comes in over the granule cells, is conveyed to Purkinje cells that can use that granule cell information to predictively adjust their spiking output, again, to synchronize some action to an event that is often carried by the climbing fiber. And the climbing fiber is thought to help the Purkinje cell to add up the granule cells in some particular way to get the desired output, in part by this canonical plasticity rule, where when the climbing fiber spikes, any of the granule cells that were active just before then can have their synapse onto this recipient Purkinje cell weakened by depression. And that has given rise to this canonical model that I've kind of summarized crudely in this image here, that there's some action you want to perform at some particular time that requires some specific pattern of Purkinje cell activity that you initially lack, which triggers an error and a climbing fiber error signal. And then the granule cells have often been thought of as providing sort of a fixed representation of the world that can be universally applied, such that this climbing fiber will selectively weaken the specific set of granule cells that will help you to produce the synchronized output from the Purkinje cell. So the thing is that it's still very unclear how you would use this elemental learning computation to meaningfully contribute to a lot of these other behaviors. And among the many questions that I think you could imagine asking, one of the ones that we've been focused on, I think one of the more concrete questions, is how does this short timescale associative learning process help you with longer timescale behaviors that involve linking things that can be separated by seconds or more? And so as one kind of baby step forward, right, we've been training animals to push for a reward that can come 1 or 2 seconds in the future, to sort of expand the length of this associative learning process. And the idea was, this canonical computation actually mainly depends on when the granule cells are active and when the climbing fibers are active. That's kind of defines classically what the Purkinje cells would learn. And so the idea was, could we observe this kind of learning process while also observing both the granule cells and the climbing fibers, which hadn't been done for any behavior, let alone sort of a longer timescale learning behavior. So, in the past, right, we've used two-photon imaging to record granule cell activity. We've also used two-photon imaging to record climbing fiber activity by way of the evoked dendritic calcium transients of Purkinje cells, which provide sort of a one-to-one readout of complex spikes and thus climbing fiber input. And so the idea was, can we sort of combine these two preps to get a simultaneous activity of these two cell types to address some of these computational questions? And the challenge is that the granule cells, 10,000 of them, provide processes that physically intersect the Purkinje cell dendrites. So you'd end up with just sort of a big fluorescent mess unless you spectrally separate them. So we use transgenic GCAMP expression, green indicator in granule cells, and viral expression of a red indicator, RCAMP2 in Purkinje cells, to get the dendritic calcium transients that correspond to climbing fiber activity. And all that remains to be solved then is the fact that the granule cell bodies are down at about 150 microns below the surface, whereas the Purkinje cell dendrites, at least where we normally image them, are around 50 microns below the surface. And so we leverage the fact that these two fluorophores require two different wavelengths, meaning two different lasers that we pass through a single objective, with one remotely focused up into the Purkinje cell dendritic layer to excite the red fluorophore, Purkinje cell dendrites, and one down in the granule cell layer to excite GCAMP in the granule cell somas. And this gave us the first simultaneous recordings from the granule cells and the climbing fibers. And I think for the purpose of this talk, the really sort of key thing about this prep, right, is that overall it's sort of local and dense sampling, meaning that we have a couple hundred granule cells that lie physically below these Purkinje cell dendrites. And what that means based on the anatomy is that the vast majority of these granule cells are providing synaptic input onto the vast majority of these Purkinje cells, meaning that at the population level, we have both the climbing fiber input and a couple hundred of the granule cell inputs onto a common set of a couple dozen Purkinje cells that we can come back to repeatedly over a week of learning. So before I talk about the imaging results, we have this learning task, right? And one of the things we keep track of is the animals licking. And what we see on novice animals is that there's a big increase in licking when the reward is delivered. Whereas when we fast forward into experts, basically the animals have already reached near peak licking by the time the reward has been delivered. So another way to quantify this is to look at the distribution of their licks across the delay period. Day one, novices ramp up their licking quickly, which then decays. Days 2 through 3, licking decays a bit less. 4 through 6, actually peak licking is towards the end of the delay, and at a week or more, really most of the licking is happening right before the reward. So another way to demonstrate more directly that the animals are actually learning the duration of this delay is to train them on a 1-second delay and then retrain them on a 2-second delay. So what I'm plotting here are only randomly interspersed omitted reward trials. So the animal doesn't get any reward, and I'm aligning them to the push. So in the 1-second experts, right, the animals push, they get to peak licking in around 1 second. The 1-second time passes with no reward delivered. The animals give up and they stop licking, whereas 2-second experts postpone their peak licking until 2 seconds, and again, wait till the reward hasn't come when they thought it was going to come, and then they ramp down their licking. So I think the animals can learn the duration of these delays. So the question, of course, that we always get is, what, if anything, does the cerebellum contribute to this? Obviously, a lot of brain areas contribute to any complicated behavior like this, so you're never going to really isolate one area uniquely. But what we did was to use these double transgenic mice that express channel rhodopsin in all Purkinje cells of the cerebellum. We exposed the same lobule 6 area where our imaging data comes from, position fiber over the window, and we can, on random trials of our choosing, sort of synchronize pulse delivery of the laser based on the movement of the robot and so forth. So here's some control data. The animal pushes here, it starts licking, these binary lick sensor contacts, the laser starts pulsing, the animal doesn't care, and so forth. When we stimulate lobule 6, animal pushes, animal starts licking, laser starts pulsing, animal stops licking, and then actually sometime later the animal starts licking again at a kind of a weird time. So here's what we see on average. First, I'm going to show you only the rewarded trials. Laser turns on only on the cyan trials. At the time of this pink box, animal stops licking. We turn off the laser, we deliver the reward. The animal easily responds to the delivery of the reward and it consumes the reward normally. So, from this, you would basically conclude that you stimulate these cells, the animal stops licking, and if you deliver the reward, the animal can immediately recover. But if you withhold the reward, you only look at omitted reward trials, you see a more disturbed pattern of licking that, after the laser turns off and we don't deliver the reward, the animal takes a lot longer to start doing anything, and what it does is kind of weird. So it's licking way later than it should be when it would normally have realized that there's no reason to be licking because it's way too late for any reward to come. And even if you use a shorter stimulation earlier in the delay period, you still see a similar kind of disturbed pattern of activity. So from this, we would generally conclude that a transient disturbance of cerebellar activity during this delay period can kind of permanently disrupt, let's say, internally driven or expectation-driven licking in the absence of some external stimulus. But of course, most of this talk is going to be about learning. So we also wanted to show that this part of the cerebellum contributes to the learning of the task. And so what we did was we trained these animals to 1-second expert status and then tried to retrain them on a 2-second delay, except on every single rewarded trial, which was 90% of trials overall, we zapped the cerebellum from the end of the delay until just after the reward was delivered. And on the remaining trials, they were sort of probe trials where there's no reward and no stim. And what we found, only looking at the probe trials, so no laser, no reward, is that basically the animals never learned to adapt the timing of their licking over the course of this 1 week to the new 2-second reward time. But if we then permanently stop stimulating, the animals can easily learn over the next couple of days to adapt their licking to the expected 2-second reward time. So I think the cerebellum is contributing something to the learning of the timing of this reward. And that's all I'm going to say about that topic, and the rest of this talk is going to be about the granule cell climbing fibers and what they might be computing that could actually contribute to this.
Mark, there was a question I wonder if we can clarify. When you say, is this stimulating the Purkinje cells? That I think I missed that. What was the
Yes, yeah, activating the Purkinje cells. Okay, so what do we see in the granule cell climbing fiber data? This is sort of all the data in 1-second delay experts. And I've sorted the cells in a way to kind of highlight, I think, an interesting finding that, a huge chunk of the granule cells seem to activate during the delay, and an even bigger chunk of the climbing fibers activate just after the delivery of the reward. So, we think this could be interesting, right? Because the animals are learning something about this delay period. We have all this granule cell activity concentrated then. So we kind of zoomed in on this, let's say 25-ish% of granule cells. And I've sorted them kind of suggestively to show you that they activate over a range of latencies. But the thing that they all generally share in common is that when the reward is delivered, for the most part, these cells turn off, and that happens to be exactly when these climbing fibers, something like 55–60% of them, spike reliably in response to the reward. But if we withhold the reward, these granule cells don't turn off, they stay on longer, and we don't, of course, get the reward-evoked climbing fiber spiking, but we get this secondary, later kind of smeared out peak of climbing fiber spiking. So we have this sort of crude idea that these granule cells are maybe kind of spanning this delay period between the action and the reward. You deliver the reward, they turn off, you don't, they stay on until the animal kind of gives up. And I'm going to be talking about them mostly as kind of representing something about the delay period. And so I think it's helpful to point out that, some alternative explanations say that they're just like a trivial readout of the movement of the tongue isn't really consistent with what we see. I mean, of course, they both ramp up during the delay, but when you deliver the reward, the cells turn off, whereas the licking gets even stronger. You withhold the reward, the cells stay on, but the licking is weaker. So it's sort of qualitatively different from sort of a trivial readout of tongue movement. And we think is more parsimoniously explained as spanning this delay for some purpose that I will give you some ideas about as we go on. So this is all in expert animals, but of course the animal doesn't know the association or the duration of delay and so forth when they're novices. So where do these signals come from? What we find is that the representation of this delay is a lot weaker in novices, but on the other hand, this climbing fiber reward response remains sort of equally strong, at least in this region of the cerebellum we've been imaging. So if the granule cells are spanning the delay, then one way to test that would be to change the duration of the delay. So again, we have 1 second experts, retrain them on a 2 second delay. And for this cohort of mice, I'm showing you the 1 second expert data just to reiterate that most of the action in the granule cells is during the 0 to 1 second period. And there's a lot less going on when the animal is actually consuming the reward. On the flip side, in 2 second experts, we have lots of granule cells spanning this longer 2 second period. So this seems to be evidence for the idea that the granule cells are somehow spanning the specific delay duration that's been learned. And again, kind of qualitatively inconsistent with the differences in licking, because of course the animals lick like crazy here. They lick a lot less here because they're waiting longer until the reward comes. They're licking like crazy here when the granule cells are off, and they're licking less here because they're still wrapping up their lick and so forth. So sort of qualitatively different. And the climbing fibers, consistent with what I showed you in the 1 second learning task, are equally strongly responding to reward in 2 second novices and 2 second experts. Okay, so it seems like these granule cells are lengthening to span the delay period over the course of learning. We know that the animals are learning the timing of the reward, so is there some relationship between these things, at least mathematically? Could you get timing information from the granule cells? And so we can crudely test this using optimal decoding, and we can show, perhaps unsurprisingly, that on individual trials, which are these thin gray lines, we can indeed track the passage of time based on population granule cell activity in experts, and we cannot do it so well in novices. And this is generally true, that the quality of timing information in granule cell populations gets increasingly good as learning progresses. So not super surprising. The interesting question is, what might Purkinje cells be doing with all this, right? And so the idea of this part of our study was, we know that this canonical plasticity rule really only depends on when the climbing fiber spikes and when the granule cells spike. And as I said, this data set is the first simultaneous observation of these two pathways onto a common set of Purkinje cells. And so can we just take the data and kind of crank it through the textbook rule and predict something about which granule cells might be providing stronger or weaker input onto a typical Purkinje cell? Meaning for every single reward, which are these pink stars, we check to see if the climbing fiber spiked. If it did, we have a putative plasticity window where we can tabulate how active every granule cell was and use that as kind of a proxy for how much plasticity or with what probability the granule cell synapses weakened onto the climbing fiber recipient Purkinje cell. And if you do this, tens and tens of thousands of trials and climbing fibers and granule cells and so forth, arguably you can get kind of a bird's eye view of which types of granule cells would be providing stronger versus weaker input onto a typical Purkinje cell. And one way, I think a nice way to visualize that is to kind of group the granule cells based on how strong we predict their synapses might be. And that's what's shown here. So each line is a group of about a couple thousand granule cells, and the color of the line tells you where it falls in the kind of synaptic weight distribution based on this prediction. And I think you see a really kind of beautiful sort of distribution of exponential or difference of exponential time constants, where cells at the bottom of this distribution peak early and then decay slowly over the remainder of the delay. And as you move up the distribution, cells' peak activity shifts progressively further to the right until you get out to these cyan curves that ramp slowly all the way until the time of the reward. And so this is sort of a temporal representation of the passage of this delay. And so you might wonder, is this really a property of the granule cells per se, or is it somehow specific to this time period when the animal really cares about the passage of time? And so if you look after the time of the reward, maybe you could have guessed this based on what I've already showed you, but all these basis elements basically collapse down into nothing after the reward is delivered by the time you get out to 2 seconds. On the other hand, if you play the exact same game in the 2 second expert data, rather than say some different set of granule cells representing the later times, all the individual basis elements are kind of stretched out to span this longer delay. And so what we think is interesting about this kind of mathematically, right, is you have to remember that this whole simulation procedure, right, only listens to the granule cell activity just before the reward when we have this climbing fiber teaching signal. And so what this is telling us is that the activity level of granule cells at the time of the reward, that alone gives us a sufficient sort of snapshot of when the granule cells were active up to 2 seconds in the past. And that's not sort of a universal property of neural activity, obviously. If you tried to do the same thing, in this data set, the 1 second expert at 2 seconds, you would fail. So this is really a specific property of granule cells that stretch out to span and link the action to the reward time. Okay, so this is a very specific set of predictions about granule cell to Purkinje cell synaptic weights that we can't actually test. I mean, we're trying, but can't really test. But what we can do is make, I guess, somewhat less specific predictions about what the implications are for Purkinje cells. So the idea is to just add up the granule cells based on the predicted synaptic strengths that I just described from this climbing fiber LTD, and to use that as a proxy for a predicted Purkinje cell output. And that's what's shown in these thin gray lines, or single trial weighted sums of granule cells with the weights determined by this climbing fiber LTD simulation. And what we predict is that Purkinje cells can basically use these granule cells to generate a long ramping simple spike rate over the course of the delay period. And the quality of that ramp, meaning how closely it tracks the passage of time, gets better and better if you look at later and later imaging sessions over the course of training. So, I mean, I think it's worth stepping back at this point and ask, what does this show? What does this not show? You might say, look, you already showed me that the granule cells acquire more and more timing information as the animal gets better at the task. So is this really telling me anything about this climbing fiber LTD-based readout, or is it really just reflecting back at me what I already know, which is that the granule cell timing information gets stronger and stronger as the animal learns? Or another way, to maybe a more dismissive way that you might put it would be, is this just like inherited from the granule cells, right? But the thing is, a Purkinje cell gets 10,000 or tens of thousands of granule cell inputs. So there's no simple or trivial way to inherit anything from a population of 10,000 inputs, right? You have to add them up in some particular way. So if you're going to posit an alternative, you have to actually describe an alternative hypothesis. So a trivial alternative would be just like averaging the granule cells or taking random weights or something like that. And if you do anything like that, you average them, you take random weights, all you get is a flat line, kind of a mess. So, and it never gets any better, even in the experts, whether you do random weights or simple average and so forth. So, while it's certainly true that your granule cells must have decent quality information if you want the Purkinje cells to produce a decent quality temporally ramping signal, that's not sufficient. So it's necessary, but not sufficient, at least mathematically. But this very kind of simplistic readout that only depends on the activity right at the time of this climbing fiber spike all the way at the end of the delay, at least mathematically, turns out to be sufficient to extract really the bulk of the available granule cell timing information into a Purkinje cell sort of ramping signal. So that's one prediction. But we can also make a prediction about how this will vary based on the duration of the delay that you've learned. So even though we get this nice ramp during the delay, we actually predict that the ramp kind of decays away after the reward is delivered, based on what we see in the granule cells, right? On the other hand, if we play the exact same game in the 2-second expert granule cell climbing fiber data, now we predict that Purkinje cells would ramp for an even longer period, right? So we predict that these ramps are specific to the duration of the delay that the animal has learned. So those are some predictions, and so we can test them by try to test them by recording from Purkinje cells, and we do this using neuropixels in kind of the standard way. And indeed, we found that in expert animals, you can identify individual Purkinje cells whose simple spike rates progressively ramp down over the course of this 1-second delay period, and after the reward has been delivered, they kind of come back up to baseline. And Purkinje cells that look like this made up something like a third of all of the Purkinje cells that we recorded from. Now, the problem with this technique, whether you think this is a big or a small fraction of the Purkinje cells, at the end of the day, electrophysiological probes are linear, and the sheet of tissue that we've been imaging is planar, and we're kind of stabbing the sheet. And so, at best, we get a couple or maybe none of our Purkinje cells from the specific area that all our imaging data has come from on the surface, dorsal surface of lobule 6. So as an alternative or complementary approach, we turn to imaging. So as many of you may know, in general, Purkinje cell somatic calcium imaging doesn't work super well because Purkinje cells have baseline tonic spike rates of up to 100 hertz, right? And that tends to saturate your calcium sensor and you can't really see anything. But if what you're looking for is a spike rate that ramps down for like 1 or 2 seconds, you would hope that something should show up in the calcium, even if it's close to saturation. And indeed, we find that if we image in this specific area where we got our climbing fiber granule cell recordings, we find that something like 80% of the Purkinje cell somas ramp down their fluorescence progressively over the course of this 1 second delay. And after the reward has been delivered, they come back up to baseline. But we also have this prediction, right, that you would ramp for longer if you learned a 2 second delay. So we retrain the animals on a 2 second delay, and indeed, now we find that, again, more than 80% of the Purkinje cells ramp down their fluorescence progressively for 2 seconds, which is very different from the 1 second case. And after the reward is delivered, again, they come back to baseline. So it seems to at least crudely bear out some of these key differences that we predicted based on the granule cell climbing fiber recordings. Okay, so I'm gonna kind of take a couple minutes to summarize and break down what I think are some of the important points. I talked about a lot of stuff, not all of which is equally important. But the headline kind of claim is that if you learn this longer timescale association, these qualitatively new granule cell temporal bases emerge, and one of their key features is that they seem to link the action to the outcome, or the reward-evoked climbing fiber spiking, up to seconds in the future. And mathematically, our claim is that this is actually an especially effective strategy if what you're trying to do is to learn these seconds-long Purkinje cell simple spiking ramps, which of course we've known for decades in the forebrain are one of the sort of most fundamental ways that neural circuits deal with long intervals between inputs, right? I'm going to break this down into a couple smaller claims. It's going to be a little bit redundant for those of you who've been listening closely, but hopefully helpful for somebody. So the first simple claim is there are granule cell temporal bases for these seconds-long intervals. So of course, people have hypothesized and theorized about granule cell temporal bases for decades with relatively little data, and I think this is very clear data for a temporal basis over a seconds-long interval. But of course, there's many different kinds of temporal bases, right? So saying there is a temporal basis is not saying much. And so what are the key features? We think the key features are that it involves many granule cells that are simultaneously spanning this interval from action to outcome, but with a range of rise and fall kinetics. And the upshot is that their activity level at the end of the interval is enough to give you a decent snapshot of their timing throughout the preceding 1 or 2 seconds. So that's the first claim, that these temporal bases sort of exist. The second claim is that they're not always there and they're not universal, right? So these things emerge with learning and they're specific to what is being learned. So the 1 second expert basis is a lot weaker in 1 second novices, and the 1 second expert basis is very different from the 2 second expert basis. In particular, the 1 second expert basis collapses back down into nothingness after the reward has been delivered over the subsequent second, whereas the 2 second expert basis spans this longer delay. So that's the second general claim, is that these bases are not universal. They emerge with learning and they're specific to what's being learned. The last claim, which is also sort of most complicated to explain, is that we think that these bases are especially well suited to learning a long Purkinje cell ramping spike rate if the main teaching signal that is available to you doesn't come until the end of the interval. And that's really, I think, a key finding of the simultaneous recordings. So look, you have an action, predict some reward 1 or 2 seconds in the future, and I'm going to kind of reason my way backwards from the goal. So if the goal is to produce a Purkinje cell ramping spike rate that spans this delay, so potentially to sort of keep track of the passage of the delay period and know how much longer you have and so forth, the usual hypotheses that are attributed to signals that look like this in the neocortex. Canonically, right, the idea behind how you would get away with a single fixed granule cell temporal basis, say the delay line basis, was that there would always be a climbing fiber teaching signal that would tell you exactly how to add up the granule cells to get whatever arbitrarily time varying function you wanted to reproduce. But that's an assumption, right? And empirically, what we find is that most of the climbing fiber activity is either at the action or at the end of the interval at the time of the outcome. And of course, we can speculate on various kinds of stories for why this is, that in reality, right, we often have scenarios where we don't get any feedback, right, between the events we're trying to link. So it may not be that easy to generate the arbitrary teaching signals that are required, say, for a delay line basis to produce such a continuous ramp. So if, as we find empirically, basically all of your teaching signal comes at the end, now it's not so trivial to produce this ramp. Now it really depends on what granule cell temporal basis you're working with. And when you view it this way, I think it makes a lot of sense that the brain generates these kind of temporally scaling, delay spanning granule cell bases, because they link, all of the basis elements are linked from the action to the reward when this teaching signal comes, in a way that allows you to produce this sort of longer timescale ramping signal in the Purkinje cell, at least mathematically. Okay, so to summarize the summary, this is sort of a picture version of the same argument. So classically, right, we had this idea of synchronizing an action before an event using a delay line basis. And you might wonder, could I do the same thing if I, instead of synchronizing an action, what I wanted to do was for a longer interval to continuously ramp? Again, something that we find all the time in the brain. And the answer is no, right? If your primary teaching signal comes at the end, you cannot use a delay line basis to produce a ramp. That's just, I mean, a pretty simple argument. On the other hand, if you have what we actually observe, which are these delay spanning ramps, now you can use that to compute, an arbitrarily long, ramping Purkinje cell signal. So given the ubiquity of these kinds of ramps and the fact that they're found in the cortex, and people over the past couple of years have also found them in Purkinje cells and the cerebellar nuclei, and they're interdependent, right? There's this recurrent effect that you shut down cortex, you shut down cerebellum, you obliterate these things in one or the other, and so forth. Given the ubiquity of these kinds of ramps, this kind of computation may be important in longer timescale behaviors. And one other kind of general claim is, we should maybe consider the possibility that different granule cell bases are used to solve different problems, in ways that maybe depend on the available teaching signals. And with that, I just want to thank everyone in the lab and collaborators that contributed to this project. And thanks to everyone for listening as well. And I'll just keep this slide up to help discussion.
Thank you so much, Mark. And it was great to see your daughter on that last picture. So, yeah, let's open it up for people to please. I know that you already had a question when you began. Let's see if that Farzana, you had asked a question. Do you want to, Farzana, do you want to?
Yes. Hi, everyone. Hi, Mark. Thanks for your great talk. Yeah, so I was curious if you have any punishments that motivates mice not to throughout the delay.
Yeah, I think, that's an interesting question. We're actually doing something a bit different. But yeah, we're trying to, address the question of how much of this general. So I think based on the data, right, these signals don't look like licking per se, but of course they do look like licking during the delay period. So you could ask, does the cerebellum generate ramps only when a ramping motor output is required, or does it generate them anytime the animal cares about the passage of an interval? And in this type of behavior, of course, as you say, it's very difficult to disambiguate. And I hope to have an answer for you, not quite with the solution that you described, but if things pan out over the next couple of months. So.
Sure, but just to understand, and you did address questions that I was going to ask, but you did not have punishment. So these mice they just lick less, or there are some trials that they don't lick at all. So that was the average trial behavior.
Correct. Yeah.
Mark, I had a question. The data that you were showing for the 1 second mice versus the 2 second mice or 2 second delay, are there the same? Do you know whether the same granule cells that were describing the bases for the 1 second, do you track them when the animal learns the 2 second one, or can you describe that transition?
Yeah, we didn't track cells in this data set. Just it was very difficult to keep track. We did come back to the same fields of view, but we didn't quite nail it. So I wouldn't want to assert that they're the same or not. But because on average across the whole population, the length of the granule cell activity profile is stretched. I'm assuming that at least some of them were shared, but I wouldn't want to assert that for sure.
Hey, Mark, super cool talk. There's something I was kind of curious more about the granule cell responses and I guess how reliable they are trial to trial. I mean, if you take an average across all the trials, like of course every granule cell is going to have some mean response and you can organize those by time, but is an individual granule cell like responding reliably around the same time across trials? Can you say
Yeah, I have
missed that?
some figure somewhere I can show you if you're curious. Give me just a second. I didn't put it in the thing, so it'll take me a second to bring up, but all right, let me see if I can. So here are a couple of those granule cells with anticipatory profiles on individual rewarded and omission trials. So you can kind of see, for example, this cell that turns off at the time of the reward but stays on longer on the omitted reward trials. So there's there's consistency. It's obviously a lot of variability, but yeah.
So do you have any sense of like like if you just took like we had a granule cell paper pretty recently, and when we took like sensory responses for, let's say, the first half of trials, and then we looked at the average response for the second half of trials and organized by the same, we didn't see
it. We did that too.
That was in like naive animals, right? So it could be totally different. What I'm getting at is super interesting.
We did that too, so it's basically the same.
Okay.
Yeah. But in terms of the actual single trial variability, that's quite a bit.
It looks pretty, yeah, they're pretty broadly responsive across the time.
During the delay, yeah.
Yeah. They don't have like a really specific time that they seem to be.
Yeah, but they all generally turn off after the reward is delivered, and they all generally turn on after the action starts. So they all kind of span that period specifically, as opposed to spanning, random periods of time.
But then this kind of makes it look like, given your plasticity rule, they're all going to be, I mean, if they're all active until the moment when the reward comes, then don't they all have the strong LTD?
So that's the argument here, is like for sure on a single trial, right? If you did this only once, given the variability, these subtle differences in activity level here are. And are you looking at my slide now, or am I still showing the other thing?
I'm looking at the heat maps, yeah.
Oh, I see. Let me go back to the. All right. So this is a cartoon, but we can go back to the actual data. But my argument is, if you did this on a single trial, of course, it's not going to give you enough information, right? Because you're talking about small differences in activity level here. But if you did this hundreds of times, or over multiple exposures, the small differences in activity level here are going to be reflected in the probability of LTD onto the Purkinje cell. And the activity levels here correspond with the times of when the cells were active. And you can see that more in the actual data as well, right? That cells at the top here ramped most slowly. Cells closer to the bottom have progressively earlier spiking ramp. So it does recover, it is recovered, but it's not recovered in one trial. And we did, of course, like subset analysis to show that basically this readout would stabilize with a couple tens of trials based on the amount of trial variability that we have.
Cool.
Farzana, you have your hand up.
Sure. Yeah, my question is more clarification, Mark. I'm sorry if I'm slow. Some of this helped, but I'm really trying to understand this model. So what you're saying is that it's not like delay line that every granule cell is active at a certain time. Instead, all granule cells are active throughout the delay. However, their peak,
Not all, but a large chunk of them, 25% or something.
25% of granule cells.
Yeah, of the active one. Yeah.
Right. And then you were saying, however, the peak timing tiles the delay.
Exactly, yeah. Or, I mean, not even the peak timing so much as the kinetics, right? So, I mean, if I compare the cyan, the slightly less cyan, and the let's say the slightly less cyan, they all peak at the time of the reward, right? But even
Yeah.
so, they have different kinetics. So
Yes.
one is
And then,
rising faster than the other.
sure. And this,
And those kinetics are recovered based on listening only to their activity level right at the time of the reward.
right. And those kinetics are learned by LTD.
No, so I don't know. I have no, well, I'm not sure what you mean by the kinetics are
Well,
learned.
that's what I
Like
mean.
they
Do
can
you
be
have-
recovered. Yes, they can be recovered by an LTD process that only depends on the activity at the time of the reward.
What do you mean
But
by
I don't know
recovery?
I have no idea what causes the granule cell profiles to assume the patterns that they do during learning.
Right. And what do you mean by recovery? You use this word. I really don't understand what you mean. What do you mean
What
that
do
they're
I mean?
recovered?
I mean, if we simulate LTD, then we predict, then the amount of LTD that a granule cell, we predict a granule cell will undergo is represented by the color here. Just simulating a textbook LTD process.
Right.
And those colors also correspond with their ramping kinetics in the previous 1 to 2 seconds. That's what we mean. If that makes sense. If it doesn't, please let me know.
And then the lobules.
Huh?
Sorry, last one, and I will stop. The lobules. Could you also talk about the different lobules that you looked
Yeah,
at?
it's a great. So, I mean, you know, the point of this study, what we were trying to do is like, can we get a handle on one specific microcircuit computation? And given the differences and how much data we needed just to get here, you know, it just we couldn't do a survey. But you know, as Court and other people have shown, right, the teaching signals vary very substantially between zones and so forth. So I think, you know, a lot more work will be needed to figure out what's going on elsewhere. In terms of the granule cells, like we have not found, we find quantitative differences in the prevalence of different profiles. We haven't found like, you know, you have ramping granule cells in one area and you don't have it a millimeter away like that we haven't really seen. But the teaching signals are a key part of this story, right? So if those are very different, as you know, Court and others have shown, then the story will be different, presumably.
The results that you showed regarding reward omission and its relationship with the granule cell activity, the key idea was that the activity remains on when the reward isn't given. And how do you, can you tell us a little bit about what you're thinking about, on the one hand, tiling of time and its relationship to the outcome that's expected, which is reward, and when it doesn't come, why does time?
So I mean, I don't have, I'll just bring up what you're talking about for anyone that, so I don't have like a hard and fast explanation for you, but I can speculate. What I think is that this is like a behavioral state, right? I am waiting for the reward. And so I'm trying to keep track of what's going on. So the reward comes, the teaching signal comes, I no longer really care about the passage of time and these cells turn off. The reward doesn't come for some period of time, which depending on how well trained the mouse is, can vary from say 300 milliseconds to 800 milliseconds or something, right? For some period of time, I will keep waiting and hoping that maybe I just didn't do a great job keeping track of time and actually the reward's still on the way. And at some point I realize there's no chance and I give up. And at that time, you know, I stop waiting, these cells turn off and I get this secondary bump of climbing fiber spiking. That's kind of our model of what's going on.
Do you think in the variety of cells that you showed, is there a rise up among even the ones that had gone up initially and had fallen, or is it just a continuation of the ones that had risen late and then they continued?
That's a good question. I don't have the plots I don't have the plots to show. We can kind of zoom in on the sorted version, which is a rough proxy. I think in general, it just looks like a slower decay of whatever they were already doing, rather than a sharp shut off that we normally see when the reward is delivered. But so one thing which I again don't have in the slide that maybe would be slightly interesting is like when in the 2-second case, when we immediately switch from 1 second to 2 second, we see, okay, let me share this here. Okay, so I don't know if you guys remember, I showed this, right, that using simulated LTD, right, we predict ramps of different durations for different conditions. And I showed that for the 1 second expert and the 2 second expert, but we also did the computation for a 2 second novice, meaning a mouse that was trained on 1 second immediately after they switched to 2 seconds. So they're very confused, right? And that's relatively similar to an omission case, I would say, at least for a couple trials, because they don't know what's going on. And what happens is these ramps saturate, right? So they stay on, but they don't continue to grow. And we interpret that as, the animal's kind of waiting, but it doesn't have access to additional time constants that would help it track the longer interval. And it's not until the animal, learns to attend to the even longer interval that instead of saturating, you have actually more ascending information coming in. That's, I don't know, maybe tangentially related to what you're asking, but yeah.
Sevgi, you've raised your hand.
Yeah, thank you, Mark. That was great talk. I wonder if you had a chance to compare this data set with any kind of classical conditioning task to dissociate the movement-related contribution.
Yeah, so we didn't do that in this paper. In the 2017 paper, we did do like play a tone that predicts a reward, and we still had ramping delays, these kind of delay ramping granule cells that had this property of reward and omission and so forth. But we didn't do a lot of it. And so, all I would say is from an existence proof standpoint that like at least some of these signals are present in both contexts. We're doing something a lot more systematic and hopefully more helpful at the moment. But yeah, that's all I have for you right now.
Thank you.
Mark, since you've been doing neuropixel recording, you must have also come across mossy fibers. Can you tell us anything about those?
Yeah, I mean, to be very honest with you, we are not like, yeah, we don't have a ton of experience with neuropixel. It was kind of like, how quickly can we churn this out to get the data back to reviewers? And personally, I am just waiting for the big consortium that Court is a part of, and for that code to come out public, and then my plan will just be to take it at face value and apply it to our future recordings, because we're not like a practiced hand at the cell identification and stuff.
Are there any other questions and comments for Mark?
I do have a question. I know I said I'm not asking. So with the remote focusing unit, is it like your 1 frame superficial, then 1 frame deep? Is it like?
No, it's simultaneous.
I see.
So one laser is focused to one depth, one is focused to the other depth, one generates red light, one generates green light, and we just split them onto 2 PMTs.
Well, thank you so much, everyone. Mark, that was marvelous. Congratulations on the paper.
Yeah, feels like a Pyrrhic victory, but we'll take what we can get. Thanks.
That was wonderful. All right, everyone. Thank you and have a wonderful day.
Thanks. Take care.
Okay.